Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing University of Hyderabad PhD entrance examination question paper for the subject biochemistry conducted in the year 2019, and I will be discussing only the biology part. So coming to the first question. The accompanying figure depicts the map of a part of E. coli genome harboring the lac operon. Lac mRNA would hybridize to which of the following DNA? So, here they have given the map of lac operon of E. coli. So, this is the laci, this is P, that is promoter, this is O operator, this is Z, Y and A, they are the structural genes. So, they are asking lac mRNA would hybridize to which of the following DNA? So lac mRNA. So from where is mRNA synthesized? So it is synthesized from the operator region, that is after the promoter. Downstream of the promoter you have the operator, lac Z, Y and A genes. So it will be synthesized from the operator to the lac A gene, up to the lac A gene. So this will be your mRNA sequence. So it will synthesize the operator, that is option 2, then lac Z, lac Y and lac A. So the answer here is option C, that is only 2, 3, 4 and 5. Then coming to the next question. A mutant of E. coli with a heat-sensitive DNA ligase. Heat-sensitive DNA ligase. Now what is ligase used for? It is used for joining DNA strands. So the ligase they are using here, that is a mutant. In mutant E. coli it is heat-sensitive. That is, it will be having enzymatic activity only at certain temperature, whereas at higher temperature than that particular temperature it will not be having the enzymatic activity. So the 25 degrees Celsius is the permissive temperature. So permissive temperature is the temperature at which this particular temperature sensitive mutant gene product takes on normal functional phenotype. That is, it will join the short strands of DNA. And 37 degrees Celsius, it is non-permissive. So it will not function at 37 degrees Celsius. And it has been used to show that lagging strand DNA synthesis is discontinuous. So they have used this particular DNA ligase to show that lagging strand DNA synthesis is discontinuous. What is discontinuous? The synthesis of a new strand of replicating DNA molecule as a series of short fragments. And these short fragments, they are joined together by DNA ligase. And it takes place on the lagging strand of the DNA. So what they have done is examination of the DNA replication in the presence of H3 thymidine, that is radio labeled thymidine in the mutant would demonstrate which of the following. So if you examine the DNA, so they have a mutant E. coli that is heat sensitive, that is having heat sensitive DNA ligase, which is working at 25 degrees Celsius and it will not work at 37 degrees Celsius. And they have used this particular heat sensitive DNA ligase to show lagging strand DNA synthesis is discontinuous. And what they have done, they have labeled the DNA with H3 thymidine and they have asked what will be would demonstrate which of the following, what will be demonstrated by this action. So accumulation of short segments of unlabeled DNA at 25 degrees Celsius and at 37 degrees Celsius. So there won't be accumulation of short segments of unlabeled DNA at 25 degrees Celsius. Why? Because 25 degrees Celsius DNA ligase will work and it will join the strands of the DNA. So it will not be accumulating as short segments of DNA. Then so that option is wrong. Then next one, accumulation of short segments of unlabeled DNA at 25 degrees Celsius but not at 37 degrees Celsius. As I told earlier, short segments will not accumulate at 25 degrees Celsius because DNA ligase will act on the short segments and join them. Then next one, accumulation of short segments of radioactive DNA at 37 degrees Celsius but not at 25 degrees Celsius. So there will of course be accumulation of short segments of radioactive DNA at 37 degrees Celsius. Why? Because 37 degrees Celsius is the non-permissive temperature, and at this particular temperature DNA ligase will not be able to act on the short segments of DNA and join them or ligate them. So they will accumulate at 37 degrees Celsius. But you will not see the accumulation of short strands of DNA at 25 degrees Celsius, because at 25 degrees Celsius DNA will be active, DNA ligase will be active, and it will cause joining of the short segments of DNA. Then next one, accumulation of short segments of radioactive DNA at 25 degrees Celsius and not at 37 degrees Celsius. So that is also not correct. So the answer is option C. So coming to the next question. During the erythrocytic stage infection of Plasmodium, the parasite undergoes repeated rounds of replication, aggression and invasion. So in the erythrocytic stage of infection of Plasmodium, it will undergo replication, then aggression and invasion. Invasion is invade to the other RBCs. So the stage specific gene expression is known to be coordinated by the functional state of chromatin. So the stage specific gene expression, that is at which stage the Plasmodium is at present, is known to be coordinated by the functional state of chromatin. State of chromatin is regulated by proteins that bind to chromatin and modify histones. So a research group wanted to understand the function of a novel protein X. So they have isolated a novel protein called as X. In silico studies, that is bioinformatics studies, showed that protein X has DNA binding domain, and it was also found to interact with other proteins involved in histone modification. So the protein X which they have isolated has a DNA binding domain. So it binds to the DNA, that may be at the promoter, we have to find out. And it was also found to interact with other proteins. So it not only had a DNA binding domain, but it also showed protein-protein interaction. That is, it was found to interact with other proteins. Which proteins? Those involved in histone modification. And these are the experimental findings. They found that conditional knockdown of protein X has no effect on parasite replication as compared to normal parasite. So if there was a conditional knockdown of protein X, knockdown means you knock down something, that is you remove or you disable the function of that particular protein. So when there was knockdown of this particular protein X, it showed no effect on parasite replication as compared to normal parasite. So after knocking out the gene, there was no effect on parasite replication. That is, the parasite replicated normally, just like the normal parasite. So what do we infer from this? We infer that the gene, that is this protein X, is not involved in replication. Okay, that is why even after knocking down protein X, it did not have any effect on replication. The next one. However, the number of infected RBCs post-aggression was less in protein X knockdown strain. So they have shown that the number of infected RBCs, that is the RBCs which are infected by the parasite post-aggression, that is after aggression. They have told that it undergoes repeated rounds of replication, aggression and invasion. So they have shown that the number of infected RBCs post-aggression was less in protein X knockdown strain. So after aggression, so it means that post-aggression means after aggression. So that means aggression is taking place even after X knockdown. Okay. So even if X protein has been knocked down, aggression is taking place. That is why they have mentioned post-aggression. The number of RBCs getting infected was less in X knockdown. So the number of RBCs getting infected was less in X knockdown. So here we can conclude that, or else we can say that, we cannot conclude, there are still more experiments to be done. So we can say that X might be involved in invasion. So X may be acting on the invasion round of Plasmodium infection. The next one. Upon knockdown of protein X, a subset of gene was upregulated in the later stages of parasite as compared to the wild type parasite line. So what did it do? The knockdown of protein X resulted in upregulation of certain genes. Then next one. Functional enrichment analysis revealed that genes which showed altered expression belong to exported and invasion family. So functional enrichment analysis or protein enrichment technique, it is a technique by which proteins of a particular type in a biological sample are concentrated for further analysis and identification. So this analysis revealed that genes which show altered expression. So genes which showed the altered expression, they have found that it resulted in altered expression of some genes post-aggression. So these altered genes, they will be mostly of invasion. I told you when I explained the second point. So this analysis revealed that genes which show altered expression belong to exported and invasion family. So they concluded that it is not, the genes are not involved in replication nor aggression, but they are involved in the invasion. Then the CHIP chip experiment protein X was found to be enriched on the promoters of gene which showed altered expression. So chip experiment, chromatin immunoprecipitation experiment, it is used to investigate interaction between proteins and DNA. So chip experiment revealed that protein X was bound to promoter of genes which showed altered expression. That is, it was bound to promoter of genes involved in invasion. Now what they have asked is, choose the best conclusion that explains the above experimental finding. So the first one, A. Protein X is essential for parasite proliferation as well as replication. So the experimental finding showed that knockdown of X has no effect on parasite replication. Remember our first point. So option A is not correct. The next one. Knocking down protein X results in defective aggression whereas invasion is normal. So knocking down did not result in defective aggression because we have already mentioned in point 2, that is, it is post-aggression. Okay, so the experiment showed that the number of RBCs infected was less in X knockdown, showing invasion genes not being expressed. The next, the above experiment could not establish any involvement of protein X in parasite aggression or invasion. The above experiment could establish involvement of protein X in parasite invasion. So C is also not correct. Then the last one, protein X enrichment to the promoter of invasion gene enhances their expression. So that is the right answer. So protein X enrichment to the promoter, that is functional, what was that? Functional enrichment analysis. So protein X enrichment to the promoter of invasion genes enhances their expression. So answer here is option D. Then next one. In mammalian cells, DNA double strand break triggers activation of the ataxia telangiectasia mutated protein kinase, okay, ATM protein kinase, which phosphorylates downstream targets that initiate cell cycle arrest, DNA repair or apoptosis. Several of these targets including P53, CHK2, BRCA1, H2AX, MRERAD-NBS that is mRNA complex are essential for DNA double-strand break repair and genomic stability. In vitro kinase assay was performed to understand the factors important in ATM activation. As a readout, phosphorylation of P53 at serine 15 was monitored using anti-phospho P53 serine 15 antibody. Okay, analyze the following Western blots. So they have told that the DNA double-strand break triggers activation of the ATM protein kinase and it phosphorylates downstream targets that will such as P53, CHK2, BRCA1, mRNA complex, etc. And these are essential for double-strand break repair and genomic stability. So they conducted an in vitro kinase assay to understand the factors important in ATM activation. So ATM is activated only when DNA double-strand break happens. So as a readout, phosphorylation of P53 at serine 15. So you have to monitor something. So for that they have done Western blotting. And phosphorylation of P53 at serine 15 was monitored using anti-phospho P53 serine 15 antibody. So this was taken as the antigen, that is P53 serine 15 was monitored using the antibody, that is anti-phospho P53 serine 15 antibody, and they have carried out the Western blot analysis and they have given the recordings over in the table. So I have brought the table over here just to answer the conclusions or explain the conclusions, okay, and we have to find out the answers. So this is the table that I have brought from there, okay. So following conclusions were drawn from the above blots. So unlike monomeric ATM, so you have got ATM monomeric over here and ATM multimers here. So unlike monomeric ATM, that is this one, ATM required both mRNA complex and DNA for activity. So in the first case, in case of ATM monomeric, okay, we will see what is happening there. ATM monomeric. So in the absence of both mRNA, that is this one, MRERAD-NBS, that is mRNA complex. In the absence of that mRNA complex and MR complex, that is MRE11-RAD50, this one, no band is present in the absence and presence of DNA. So this is DNA here. So in the absence of DNA, you don't get a band over here. Even in the presence of DNA, you don't get a band over here. In the absence of mRNA and MR in case of ATM monomeric. But in the presence of mRNA complex, that is over here, plus, they have given a plus there, mRNA complex, and in the absence of MR complex, over here, you get a band, see. For both absence and presence of DNA, you get a band over here. That is, a Western blot has given a band. Showing that mRNA complex is required by ATM monomeric and its activity is not affected either by the presence or absence of DNA. So only when mRNA complex was present, ATM monomeric showed bands, but it did not depend on whether DNA was present or DNA was absent. It gave bands. So mRNA complex is required for ATM monomeric. But it doesn't require the presence of DNA. Now, in the absence of mRNA, that is over this column, that is in the absence of mRNA and the presence of MR. Okay, here ATM monomeric activity was observed even in the presence and absence of DNA. So even if mRNA was not present and if MR was present, whether DNA was present or absent, ATM monomeric showed activity, that is, it gave two bands. So in case of MRN, if N and here MR, that is N is absent here, that is NBS1 is absent in the case of MR11, RAD50, that is over here. So NBS is not that important for ATM monomeric activity. That is why we could see bands even with only MR being present and DNA absent or present. Then next one, ATM multimeric case. So in the absence of both DNA and mRNA complex, that is over here, DNA is absent, mRNA complex is also absent, then ATM, no bands are observed in the presence of DNA. No bands were observed. That is over here, you don't get any bands. Now we will see in the presence of DNA and mRNA complex, that is DNA, and this is the mRNA complex in the presence. Band was observed. Bands were observed. But how are the bands? The intensity of band was increased with increasing DNA concentration. That is 2.5 gave a slightly thinner band than 10 nanogram of DNA, or 40 microgram of nanogram of DNA gave a much thicker band. So the band intensity was increased with increasing DNA concentration. But band was observed when only DNA was present, even at higher concentration, and mRNA was absent. So here we didn't find, so even if you have 40 nanogram of DNA, but if mRNA is absent, you don't see a band over here even if the DNA concentration was high. Showing that both DNA and mRNA are required for ATM multimeric. That is, it requires both DNA and mRNA for its activity. And here also, if DNA is absent and mRNA is present, no band is observed. But if DNA is present at 40 nanograms and if mRNA is not present, then also no band, showing that mRNA and DNA are required for ATM multimeric. So first option is first conclusion is, unlike monomeric ATM, multimeric ATM required both mRNA complex and DNA for activity. So that is correct. Okay. Then NBS is not that essential for ATM activation in vitro. That I already discussed when I discussed ATM monomeric. See here only M and R are there. See here, M and R are present, but mRN is not present. Then DNA is absent or DNA is present, you get a band. So N is not essential for ATM activation in vitro. So 2 is also correct. Then next one, activation of ATM is mRNA independent. No, it is dependent. Only if mRNA is present, you get bands over there. Yes, in case of ATM monomeric as well as in case of ATM multimeric. So that is not correct. With increase in DNA concentration, the activity of ATM increases. That is also correct. So the answer here is option 1, 2 and 4, that is option D is correct. Then next one. So before coming to the next question, I would like to know you all to know what is co-immunoprecipitation. So co-immunoprecipitation, it is a technique for identifying protein-protein interaction, and according to this, protein bound to another protein will be eluted out together. That is, if protein A is bound to B, they both will be eluted together in the case of co-immunoprecipitation. Then the next term is pull down assays. Pull down is just like you pull it down. So if you are trying to precipitate A out of here, A will, or else you are centrifuging this particular protein complex, that is A and B together. And what will happen is, along with A, B also will be pelleted out. Okay, because A is pulling down B along with it, and so they both will be present together as a complex in the pellet. Okay, so that is what is called as pull down. So A is pulling down B. And co-immunoprecipitation and pull down assay are closely related methods to identify stable protein-protein interaction. And in a case of co-immunoprecipitation, an antibody-bound protein is itself bound to an other protein that does not bind with the antibody. That is, you have got two proteins A and B. For you have generated antibodies against protein A, and these antibodies, they will not bind to B. Okay, but A and B, they both will be um, sub, precipitated out. Okay. And this particular protein complex you can study by using this co-immunoprecipitation technique. So coming to the question. So it was observed that when a molecular chaperone C is inhibited, the cells become sensitive to DNA damaging agent. So if C is present, cells will not become sensitive to DNA damage. But if C is absent or inhibited, the cells become sensitive to DNA damaging agent. To understand this mechanism, a series of experiments were performed. Cells were treated with DNA damaging agent and two genes P and Q were found to be upregulated. So to understand the mechanism, they performed a series of experiments. So they treated the cells with DNA damaging agent and they found that two genes, that is P and Q, were found to be upregulated. Then they used yeast 2 hybrid assay. This is for yeast 2 hybrid assay is for performing protein-protein interactions. So yeast 2 hybrid assay was performed to check for the interaction of P and Q with chaperone C. So they performed yeast 2 hybrid assay and to check the interaction of P and Q with the chaperone C. And only P was found to interact with C. Q didn't interact with C, only P was found to interact with C. Then co-immunoprecipitation was further performed to study interaction. Given below is the picture of the Western blot obtained. So this is the picture of the Western blot obtained. This is anti-C pull down. That is, I think it is over here. That is in the second lane. The graph is, the picture is not clear to me. In the question paper also it was not that clear. But anyway, I take it for being in the lane 2. The next one, RT-PCR analysis revealed no effect on transcription level of gene P and Q upon inhibition of chaperone C. So even if chaperone C is inhibited or chaperone C is inhibited, you get transcription level for genes P and Q. The next one, in order to determine whether P or Q are the client of chaperone C, that is whether P or Q is the one that binds to the chaperone C, they conducted the steady state level of proteins P and Q were determined in the presence of two inhibitors. So one that inhibits chaperone C activity and another that inhibits proteasome mediated degradation. So one is that which inhibits chaperone C, C inhibitor in the presence as well as proteasome inhibitor. It is established that if C is inhibited, any of its client would degrade via proteasomal pathway. That is, if C is inhibited, this C is inhibited, then any of its clients, that is those which bind to C. So here they have already told that P was found to interact with C with yeast 2 hybrid assay. So what they are trying to say is, if C is inhibited, any of its clients would degrade via proteasomal pathway. So if you inhibit or if you add the C inhibitor and if C is bound to P, P will be degraded via proteasome mediated pathway. So, okay, so I have carried over the picture over here for explanation. Okay. Okay. So here is the options they have given. On the basis of above experiments, following statements were made. State which one is correct. Gene P and Q function as complex and both are client of C. They both are not client of C. As per yeast 2 hybrid assay, they have found that it is P which is a client of C. Okay. And further experiments they have conducted to establish the fact that P itself is the client of C and Q is not client of C. Okay, then next one. P and Q do not form a complex and only gene P is client of C. P and Q, they do form a complex. I will explain now. Wait, we will see option C also. Wait, now before that, okay, I will explain P and Q, they form a complex. Okay, okay. So here, that is input. First one, input, lane 1. Input showed all the bands in the lane 1. That is with anti-C, anti-P, anti-Q, you got all the 3 bands. In lane 2, that is over here, that is 2, pull down was performed. Pull down of C was performed. So anti-C pull down. I think it is in lane 2 only. Okay, so lane 2, pull down of C was performed. So P, as it is bound to C, the 2 lane is of the pellet. Okay, so P as it is bound to C, so we know that as per the yeast hybrid assay that P is bound to C. It also settled down in the pellet because C pulled it down, pulled it along with it. So it also got settled in the pellet. And since Q is bound to P, it co-precipitated along with P bound to C. So P, it is bound to C and Q, it is bound to P. But it is not bound to C. Since Q is bound to P, it co-precipitated along with P bound to C. So that is why you got a band over here in case of anti-Q. Okay, lane 2. Lane 2 is the pellet fraction and you got a band against Q. Okay, so P and Q form a complex and a third band was observed. So P and Q, they do form a complex. If you take lane 3, that is supernatant, you find a band of Q over there, that is unbound Q. That is Q which didn't form a complex with P. Those were observed in the supernatant, that is lane 3. Okay, then next one. Next one. In order to determine whether P or Q are the client of chaperone C, the steady state level of proteins P and Q were determined in the presence of 2 inhibitors. One that inhibits chaperone C activity and another that inhibits proteasome mediated degradation. It is established that if C is inhibited, any of its clients would degrade via the proteasomal pathway. The plot of such an experiment is shown below. So this is the Western blot. This is plus minus means if in the presence of C inhibitor and minus means in the absence of C inhibitor. Similarly, if in the presence of proteasome inhibitor is plus and proteasome inhibitor is minus in the absence of proteasome inhibitor. Now this anti-actin, it is just a control. Okay, so it will be present in all 3 cases. It is control. So first one, in the absence of C and proteasome inhibitor, this is the lane 1. So you don't have a C inhibitor over there nor a proteasome inhibitor. So C is not inhibited. It will be bound to P and P will form complex with Q. So you will get 2 bands over here and they are present. Then bands were observed for P and Q. Okay. Then in the presence of C inhibitor, that is plus in the case of C inhibitor and in the absence of proteasome inhibitor. So proteasome activity will take place. Okay, that is why proteasome inhibitor is not added. And what they have told in the question is One that inhibits, okay, chaperone C activity and another that inhibit proteasome mediated degradation. It is established that if C is inhibited, any of its client would degrade. Okay, so if C is inhibited, so here we have added the C inhibitor in the second lane. So that means C is inhibited. And what will happen to its client, that is P? It will get degraded via the proteasomal pathway. Proteasome inhibitor is not added and P will get degraded via proteasome pathway. That is why you didn't find a band or you got only a very faint band for P in lane 2. And since Q is not a client of C, you got a band similar to that of the normal case. That is minus minus. Okay, that is a normal case. So since Q is not an client of C. Okay, so Q is not a client of C. You got a band over here. Then next one. Here, when both the C inhibitor and the proteasome inhibitor are present. So C inhibitor is present. So naturally, P has to get degraded via proteasomal pathway. But in this case, proteasome inhibitor is also present. So proteasomal pathway will not be taking place because it is inhibited by proteasome inhibitor. So P will give a band over there. So you got a band over here and a Q will naturally give because it is not a client of C. So it will give a band over here and you will get two bands for, that is for P and Q in the presence of C inhibitor and proteasome inhibitor. So with this experiment we can conclude that P itself is the client of C and P and Q form a complex. So what is the answer? Gene P and Q function as a complex and both are client of C. No, only P is client of C. Then P and Q do not form a complex and only gene P is client of C. P and Q, they do form a complex. That is why you got a band in first Western blot. Then gene P and Q form a complex and only P is client of C. That is the correct answer. So answer here is option C. Then next one. In an organism, expression of few genes were found to be altered when external stress of heat was given. Given below is the CT value obtained through real-time RT-PCR for genes involved in a pathway under control and experimental conditions. So you know what is CT value and RT-PCR after hearing all the things about corona in newspaper. So I am not going to explain all that. Okay, to get the molecular insight of the phenomenon, chromatin immunoprecipitation was performed in control and heat stress condition using anti-T antibody and IgG which served as negative control. Given below is the gel picture of PCR done using primer specific to R promoter from IPDNA of above mentioned antibodies. Now actually the gel picture, that is the blot picture. Oh, sorry. It was not clear. Okay, you can see. This is the picture what they have given, and it is not clear. So, what I will do is I will explain using the table. In T knockout background, no transcription alteration of both V and R was observed. On the basis of above experiments, following statements were made. State which one is correct. Okay, so upon heat stress, V and T are upregulated twofold, R is downregulated. So we will. So this is your table. The table is smaller, but I hope you will be able to see it. Okay, because I have to put this photograph over here, so I have to make the table small. So no other option. Okay, now how to calculate this fold? down regulated or up regulated, etc. we can see over here. So the first one, normalize CT of the target gene to the CT of the reference gene. That is the first step you have to do. CT of the target gene, that is for heat stress, and CT of the reference gene. Okay, that you have to normalize. So delta CT calibrator, that is equal to CT target cell minus CT reference cell. This calibrator is nothing but CT control, and your housekeeping genes are the control, are acting as control. So delta CT control is CT target cell, that is heat stress. So that is 17, and CT reference, this is the CT value for control, that is 15. So you just put it in the formula, 17 minus 15 is equal to 2. Then delta CT test, that is VR and T. So CT target minus CT reference, the same formula, but we will be taking for VR and T. So coming to the first one, that is V gene. So it is 14 minus 13. This is the target 14, and this is the reference, that is 13. So 14 minus 13 is equal to 1. Then R, that is 26 minus 21 is equal to 5. Then for the T gene, it is 18 minus 17, that is equal to 1. Then you have to find out the delta CT value. Normalize delta CT of test sample to the delta CT of the calibrator. So first you have to do for the V gene. Okay, delta CT value for the V gene. So delta CT of the test minus delta CT of calibrator. So we already have the delta CT of calibrator over here, that is 2. Now for the V gene, delta CT of the test is 1. So 1 minus 2 is minus 1. Next is for the R gene, that is delta CT, that is 5. For R gene, delta CT test value is 5, and for the reference gene, that is 2. So you get 5 minus 2, that is 3. For the T gene, delta CT value is 1 minus 2, that is minus 1. Then next what you have to do is calculate the fold difference in expression. So that is given by formula 2 raised to minus delta CT. So for the V gene, 2 raised to minus of minus 1. Delta CT for V gene is minus 1. That is 2 raised to 1, that is 2. Then for R is 2 raised to minus delta CT. So answer is 2 raised to minus 3. So that will be around 1 by 8. And for T, that is 2 raised to minus delta CT. That is 2 raised to minus of minus 1. That will be 2. So we find that upon heat stress, V and T are upregulated. Upon heat stress, V gene and T gene are upregulated. They are positive values, whereas the R gene is downregulated 8-fold. It is a negative value, 2 raised to minus 3. So it is 1 by 8. It is downregulated, whereas the B gene is upregulated and the T gene is also upregulated 2-fold each. That is why you got the answer 2, whereas this one R gene is downregulated 8-fold. Okay, so that is the answer. Upon heat stress, V and T are upregulated 2-fold, while R is downregulated 8-fold. Then coming to the next question. Okay. The following data was obtained from a transduction experiment. So this is the donor, tryptophan plus, leucine plus, arginine minus, and recipient tryptophan and leucine are not present but arginine is present. Among the tryptophan transductants, the researcher obtained the following phenotype. So leucine plus arginine plus is 48, leucine plus arginine minus is 24, leucine minus arginine plus is 144, and leucine minus arginine minus is 3. the order of the loci is most likely to be. So here we see that leucine minus arginine plus has the maximum number of transductants. So it will be near to the entry point of the gene and the first one to pass into the recipient. Or I can say the first one to pass the more number of times into the recipient cell because it is having the maximum number. So arginine will be the first one. So first one will be arginine. Then next higher value is for leucine plus arginine plus. So leucine comes next to arginine. And the next gene will be TRP. So the answer here is arginine, leucine, tryptophan. Answer is option B. Then next one. If a double crossover occurs in a paracentric inversion, the four meiotic products are likely to be whether they will be inviable, viable, or only non-recombinants are viable, dicentric chromosome. So what is paracentric inversion? The one in which the inverted segment does not include the centromere. So if you have got a gene, the chromosome, the centromere is not involved. Only those genes on either side of the centromere, they will undergo inversion, but without including the centromere. Okay. So what will happen is they will be viable. Okay. The double crossover occur in paracentric inversion. The meiotic products are likely to be viable. Why? Because all genes are present. Only inversion has taken place. That is, ABC has been inverted and kept as CBA. Okay, ABC. If you take ABC gene, it is inverted and kept as CBA, but the genes are present. Nothing is happening with the genes. So both the normal and inverted, because all genes are present in the normal and inverted chromatids. So there is no deletion or duplication taking place. So all the meiotic products are likely to be viable. So answer is option B. Then next one. Two bacterial strains, one isolated from sugar syrup and the other from soil, utilized glucose as the sole carbon source. The kinetic parameters of the enzymes from these bacteria that use glucose as substrate are given below. So they have given the Km and the Vmax value for syrup as well as for the soil. And the enzymes isolated from these two bacterial strains were then assayed in three different concentrations of glucose, that is at 10 millimolar, 100 millimolar and 1000 millimolar glucose. Which of the following statement is correct? Strain B soil will have higher V0 at 100 millimolar glucose than B syrup. Strain B syrup will have higher V0 at 100 millimolar glucose than B soil. Strain B soil will have higher V0 at 10 millimolar glucose than B syrup, and strain B soil will have higher V0 at 1000 millimolar glucose than B syrup. So in total what you have to conclude is you have to find out the V0 value at different substrate concentrations. So here it is, I have carried over the graph as well as the options the table as well as the options here, and I have put the photographs for the calculation. So the first case is B syrup. So V0 is equal to, as per Michaelis-Menten kinetics, you have Vmax concentration of S by Km plus concentration of S we will see for 100 millimolar glucose. So B syrup. So substitute, you have got Vmax of 1000 in the case of B syrup and Km value is also 1000. Substrate concentration is 100 millimolar. Substitute and you will get 90.9. If it is B soil, you will get 90.90. Now you calculate at substrate concentration 10 millimolar. So V0 for B syrup is 9.90 and B soil is 50. At 1000 millimolar, it is B sugar at 500. B syrup is 500 and B soil is 99. So we will see the options. Strain B soil will have higher V0 at 100 millimolar glucose than B syrup. So what is B soil at 100 millimolar glucose? That is 90.9, and B syrup at 100 millimolar glucose is 90.9. So naturally that is wrong. Both are equal. Both are 90.9. So A is not correct. Then next one. Strain B syrup will have higher V0 at 100 millimolar glucose than B soil. That is also not correct. Then strain B soil will have higher V0 at 10 millimolar glucose than B syrup. So strain B soil, this is B soil and this is B syrup at 10 millimolar substrate concentration. B soil is 50 and B0 is 9. So what they have told, strain B soil will have higher V0. Yes, B soil is having 50 and B syrup is having 9.9. So option C is correct. Then next one. A sequence of amino acids is in a certain protein is found to be serine, glycine, proline, glycine. The sequence is most probably part of an. So glycine, proline residues, they are found in the turns. So the answer here is option D. Then next one. Which of the following statements about nuclear transport is incorrect? Mark the most appropriate answer. So protein destined to nucleus contains signal sequences. That is correct. Small proteins can freely enter the nucleus. Yes. Then next one. Signal sequence is removed after transport into nucleus. Now nuclear localization signal, that is lysine, lysine, arginine, lysine, that is a signal sequence, is present inside the protein. That is, it is near to the N-terminal end of the protein but not at the N-terminal end. Okay, the signal sequence is near to the N-terminal end of the protein and it is not at the N-terminal end. Since nuclear localization signal is an internal localization signal, it is not removed by specific peptidases. So the answer here is, they have asked which is incorrect. So the answer is option C, that signal sequence is removed after transport into nucleus. Then next one. Addition of N-linked oligosaccharide is done. So in endoplasmic reticulum and then modified in the Golgi. So that is where N-linked oligosaccharides addition are done. Answer is B. Then next one. In a 3-point test cross, the most frequent class of progeny are A, B and small c, and small a, small b and capital C. And the low frequency are capital A, small b, capital C and small a, capital B, small c. Which allele is in the middle? So they have given a 3-point test cross and we can see that these two, ABC and this ABC. Okay, this ABC and this ABC, they are the parentals because they are the most frequent. And these two, that is ABC, these two, okay, ABC, these two, they are found at low frequency and they are double crossovers. The next important point is that in a double crossover event, moves the middle allele from one sister chromatid to the other. And what they have asked in the question, which is the middle allele. So these are the parentals and these are the double crossovers. If you see them, you see that B capital C. So B capital C, this region is remaining constant, whereas B small c, that is B small c, that region is also remaining constant without any change. But A is moving this way and that way. A is moving from here to here and here to here. So A is in the middle. Okay, that's how you conclude it. So the answer is option 32, that is capital A, small a. Then next one. Which of the following statements about cytoplasmic inheritance is incorrect? Uniparental inheritance. Yes, it is maternal inheritance. Cytoplasmic inheritance is for mitochondria and chloroplast. Okay. So it is uniparental inheritance. Yes, it is maternally inherited. They show non-Mendelian segregation. Of course, that is also correct. Progeny may have different degree of heteroplasmy. So what is heteroplasmy? Heteroplasmy is the presence of more than one type of organellar genome within a cell or an individual. Organellar genome means either from mitochondria or chloroplast. There can be more than one type, that is different types of organellar genomes within a single cell or an individual. Now, how does this arise? This arises because eukaryotic cells contain many hundreds of mitochondria. If we are taking the case of mitochondria, they contain many hundreds and thousands of mitochondria with hundreds of copies of mitochondrial DNA. So it is common for mutation to affect only some mitochondria, leaving the others unaffected. So that is the how heteroplasmy arises. And organelle genes in heteroplasmic cells can segregate because they each have several copies of their genome. This may result in daughter cell with differential proportion of organellar genotype. So progeny with different degrees of heteroplasmy, that is also correct. So what is the wrong statement? Can be mapped relative to nuclear genes. So answer is option D. Then next one. Genome imprinting is. So in genomic imprinting, the ability of a gene to be expressed depend on the sex of the parent who passed on the gene. So in some cases, imprinted genes are expressed when they are inherited from the mother, and in other cases they are expressed when inherited from the father. So genome imprinting is option C, that is parent of origin dependent expression of genes. Answer is option C. Then next one. Which protein is involved in the separation of the two interlinked daughter chromosomes when DNA replication is terminated in E. coli? So the double helical nature of DNA and its semi-conservative mode of replication causes two newly replicated strands to be interlinked with each other. Now these things, they have to be removed for the chromosomes to get segregated into the daughter cells, so that cell division can be completed. So which protein is involved? It is topoisomerase 4. Answer is option C. Next, what would happen if the concentration of dideoxynucleotides was too high in chain termination sequencing reaction? Mark the most appropriate answer. So you know what is chain termination reaction sequencing method, and you use dideoxynucleotides. Now what will happen if the dideoxynucleotides are too high? So if you increase the DDNTP, that is dideoxynucleotides, you would get more sequence close to the primer, but make it more difficult to read the sequences 200 to 300 nucleotides further down. Why? Because the concentration of DDNTP is more. They will keep on adding, and you will get the sequence very close to the primer. You won't get the whole stretch of your DNA sequence because they are more in number. So you would get more sequence, more sequence close to the primer. Okay, but make it more difficult to read the sequence 200 to 300 nucleotides further down. So the answer here is the reaction would produce very short molecules. Answer is option B. Which of the following statements about REC-A, a protein involved in homologous recombination in E. coli, is wrong? It hydrolyzes ATP to promote branch migration. Yes. It binds to single-stranded DNA to form a filament. Yes. It is an ATP-dependent nuclease that generates single-stranded DNA. It is a DNA dependent ATPase. So the answer is option C. The others are correct. Now next one. Which of the following immune cells is represented if the immune cell is CD45 CD3 is cytotoxic, can kill virus infected cells and may also interact with lipid or glycolipid antigen presented by CD1 molecules. So these characteristics, it is of natural killer cells and killer T cells. Okay. Natural killer T cells. So answer is option B. Then next one. Which of the following statement is correct for MHC class 3 genes? Encode glycoproteins expressed on the surface of nearly all nucleated cells. So all nucleated cells expressing this particular glycoprotein, it is true for MHC class 1, not class 3. Then next one. Encode beta 2 microglobulin gene that form part of the MHC class 1 molecule. So option B, it is for class 1 molecule. Then next one. Encode glycoprotein that primarily present exogenous antigenic peptides to CD4 plus T cells. So that is the case with MHC class 2, not with class 3. So the answer is option D, that is encode components of the complement system. Then next one. Which of the following mechanism does not contribute to the generation of diversity of T cell receptors when compared to immunoglobulin diversity? So it is somatic hypermutation. So somatic hypermutation occur in antigen activated germinal center B cells, not T cells. T cells, there is no somatic hypermutation. And somatic hypermutation does not occur in T cell receptor genes, so that variability of the CDR1 and CDR2 region is limited to the germline V gene segments. So the answer is option A, that is somatic hypermutation. Then next one. A patient suffering from anemia goes to a doctor. A blood test revealed vitamin B12 deficiency and circulating antibodies directed against gastric parietal cell. Hyposecretion of which of the following gastric secretory products is responsible for this case of anemia? So its answer is option D, that is intrinsic factor. Now intrinsic factor is a protein made in the stomach and it is needed to absorb vitamin B12. So lack of intrinsic factor causes vitamin B12 deficiency, anemia, and it is pernicious anemia. So the answer is option D, that is intrinsic factor. Then next one. Which of the following enzyme has bound thymine pyrophosphate, TPP? So it is for pyruvate decarboxylase. Answer is option A. Tandem enzymes have two different activities on the same polypeptide chain. Which of the answer is correct? So tandem enzymes is a pair of opposing enzymic activities that are resident in the same polypeptide chain. So that can be seen in the case of phosphofructokinase 2 and adenylate transferase. Answer is option B. Then next one. Glutamine serves as an amide group donor for the biosynthesis of which of the following? Mark the most appropriate answer. So it is for option A, that is histidine, tryptophan, glucosamine 6-phosphate and nucleotides. Then next one. Which of the following statement is correct? N-linked glycan core oligosaccharide is formed on dolicol pyrophosphate sequentially and contain N-acetylglucosamine, mannose, glucose residues linked to asparagine residue in proteins. Now that itself is correct. So I am not reading the others. So answer is option A. Then which of the following statement is correct? So UDP GLC NAC, that is N-acetylglucosamine 1 phosphate. Okay. GLC-NAC is N-acetylglucosamine 1-phosphate phosphotransferase absence leads to non-phosphorylation of mannose residues on lysosomal enzyme and causes eye cell disease. So eye cell disease, it is an autosomal recessive disorder caused by the deficiency of this particular, that is GLC-NAC phosphotransferase. That is N-acetylglucosamine 1-phosphate phosphotransferase, and it phosphorylates mannose residues to mannose 6-phosphate on N-linked glycoproteins in the Golgi apparatus. And without this mannose 6-phosphate, they won't be targeted to the lysosomes, and erroneously they will be transported from Golgi to the extracellular space. So consequently, lysosomes lack the requisite hydrolytic enzyme needed for catabolism of cellular debris. So the debris accumulates within the lysosome and form the intracellular inclusion. Hence the name eye disease for this particular disorder. So which statement is correct? Option A is correct, whereas in option D it is GAL, not GLC. So option B is wrong, and option C is also wrong, and arginine mutated to alanine, that is also not correct. So option A is correct. Now next one. After plotting lean beaver Burke plot of the protein of the kinetic data of an enzyme, its slope was found to be 0.095. The plot intercepts the y-axis at this value and calculate the Km of the enzyme. So this is the lean beaver Burke plot, 1 by V, 1 by concentration of substrate, that is 1 by concentration of S. And the x-intercept, that is this one, this is minus 1 by Km, and this is the y-intercept, and it is 1 by Vmax, and Km by Vmax is your slope. Now just substitute everything and you will get the answer. So Vmax is equal to 1 by 0.0126, that is 79.37 into 10 raised to minus 6. And Km by Vmax is equal to 0.095. Substitute the value of Vmax over here and then find the Km value. And you will get 7.54 into 10 raised to minus 3 molar. So the answer is option B. So coming to the next question. Which of the following amino acid cannot act as a nucleophile in the enzyme catalysis? So it is asparagine. Answer is option D. Then, which of the following methods cannot be used to determine the molecular weight of an enzyme? So size exclusion chromatography, yes. Ultracentrifugation, yes. Biuret method, no. So answer is option C. And SDS page also you can use to determine the molecular weight. Then next one. Which of the following tools you will use to align distant pair of amino acid sequences? So for that you will be using PHI-BLAST. PHI-BLAST. Answer is option B. Then next one. Family relationships can be found in the following database. So ProCyte, it is a protein database and it consists of entries describing the protein families, domains and functional sites as well as amino acid patterns and profiles in them. So the answer is option B, that is ProCyte. The next one. If succinate is the substrate, how many H positive ions would have translocated across the inner membrane of mitochondria via the Q cycle redox loop mechanism? So transfer of first electron results in the free radical semiquinone form of the Q, and transfer of second electron reduces the semiquinone form to ubiquinol form. And during this process, 4 protons are translocated from the mitochondrial matrix to the intermembrane space. Now, then the second protein complex in the electron transport chain is succinate oxidoreductase, and this protein contains the enzyme succinate dehydrogenase, and this catalyzes the conversion of succinate to fumarate, and in the process FAD is converted to FADH2, and this energetic FADH2 give off two energic electrons to the iron sulfur chain, or clusters, that ultimately transfer them to coenzyme Q. But complex 2, that is succinate Q oxidoreductase, it does not pump any hydrogens to the intermembrane space. So whatever protons are pumped, it is from the Q. So the answer is option C, that is 4 protons. Then next one. When radioactive H3NADH is added to a cell extract containing mitochondria, radioactivity quickly appears in the mitochondrial matrix. However, when C14NADH is added to the same cell extract, no radioactivity is found in the mitochondrial matrix. This is because the radioactive H3 is transported from the membrane via shuttle system, whereas C14 cannot pass through the membrane. Okay, so that is the answer, that is option C. That is, ions are not passed through the membrane of mitochondria, but shuttle system transport H positive ions. So answer is option C. The next one. How long does it take to synthesize a protein of molecular weight 50,000 at 37 degrees Celsius? So 400 amino acids can be synthesized in 20 seconds, and one amino acid is 110 Daltons. So 400 amino acids will be 44,000 Daltons. So 44,000 Daltons in 20 seconds. That is okay. So they have asked for molecular weight of 50,000. So 50,000 Daltons in 50,000 into 20 divided by 44,000. So that will be 22.72 seconds. That will approximately equal to option B, that is 37 seconds. Now don't worry that you didn't get the correct answer, because 400 amino acids in 20 seconds can vary. That number can vary. From the book I referred, it is 400 amino acids in 20 seconds. They can be even 500 amino acids in 20 seconds. Okay, now, okay. Now next one. 4 genes KYUA, KYUB, KYUC and KYUQ are required to synthesize substance Q from P in the reaction sequence P giving B, giving C, giving A, giving Q. Each of these biochemical reactions can be detected. The product of the gene KYUA is needed to synthesize substance A from C. Okay, so this is the reaction sequence, that is P giving B, giving C, C giving A and A giving Q. Now the product of gene KYUA is needed to synthesize substance A from C. So A is being synthesized from C using the product of the gene KYUA. Similarly, product of gene KYUB is need to synthesize B from P. So B is synthesized from P by action of KYUB. Product of gene KYUC is needed to synthesize C from B. So C is synthesized from B by action of enzyme KYUC. And product of the gene KYUQ is needed to synthesize Q from A. So this is Q and this is A. So Q is synthesized from A by action of gene KYUQ. Now addition of radio labeled C14P yields C14Q. So C14P, it will give rise to C14Q. If you radio label C14P, you get radio labeled C14Q product. A mutant is found in which addition of C14P yields C14A but not C14Q. So if you radio label C14P for the mutant, you will get only C14A but you will not get C14Q. So up till A it is synthesizing. Beyond A it is not synthesizing. So in what gene is the mutation? It is KYUQ, because KYUQ is necessary for synthesizing Q from A. So the answer here is option D, that is KYUQ. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.